आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सॉल्यूशन पाओ। डाउनलोड नाउ।
So here we have to illustrate the design of a bioreactor, and also we have to highlight the difference between the flask and that we use in our laboratory and also a bioreactor. So first we discuss about the design of a bioreactor. So what is a bioreactor? Bioreactors are the industrial setup where we are using or generating the enzymes or the proteins with the help of the organisms or microorganisms. So that is why the name given as the bioreactor. The reactor is designed such a way that to produce the proteins or enzymes with the help of the organisms. Those are called as the bioreactor. Okay. So now let us now come to the parts of the bioreactor and its construction. So here it has a motor. This is a big bioreactor which we are using in industry. And it has a motor. These motors are fitted with a flat blade-like structures, and also these flat blade-like structures are helpful in the churning or the impelling the liquid which we have brought. Okay. So here first of all, I'll just read out the parts of the bioreactor and then come one by one. This is a motor, foam breaker, flat blade impeller, cultural broth, sterile air, sterile for steam for sterilization, and acid or pH base for the control. So what is a motor? It is used to run the blades. Okay, here. And here there is a flat blade impeller. So where we in this reactor we are adding the cultural broth. What is a cultural broth then? So cultural broth refers to the nutrient solution okay, so where it contains all the nutrients required for the growth of the microorganisms. Okay, so then what it will do, these cultural broth, these impeller or the blades which are with the help of the motor, it will run throughout the process such that these cultural broth is equally distributed among the microorganisms present here. That is the main advantages of this impeller here. And there is a below you can see there is sterile air. So this sterile air is passed through this inlet in order to for the air circulation during the process. So this sterile air reduces the contamination. The sterile air reduces the contamination. Okay, reduces contamination during the process. And then we have a steam for the sterilization. So whenever required for during before or after using, there is an inlet pipe here through which we can pass the steam to sterile the whole process or whole system. And each bacteria or the fungi will require a specific acid or pH for their growth. So therefore there is a acidic and pH base pH controllers are there to control the acidic or the particular pH. Say for example, fungi very well grow in the pH of the 5.6, whereas bacteria grow in the 7 pH. So there is a controller knob here where you can control the acid and the pH base. So here this the main advantages of this bioreactor is that the process is continuous. That means there is a continuous production and also the continuous removal of the finished products happens here as there is this process is happening in a controlled manner. There is a downstream process, that is removal of the finished product, that is proteins or the enzymes. There is a continuous process because we also have a outlet here. So we have outlet. Since we have a outlet here, so what we are doing, we are downstreaming the process of the protein and the enzyme removal. Okay, so that is the main advantages here, and there is no re-inoculation further. So there is no need of re-inoculation again and again since the process is done in a controlled and sterilized manner. So if we come to the laboratory, so see here we can see the laboratory. In the laboratory we have a conical flask here. So here we can consider this as a conical flask. So you can see here in this condition there is a controlling of the process is happening in this here. So here what will happen, in this conical flask also there is a broth here. We can say this is a liquid culture, this is a broth here, and so we are already pre-inoculated the organism and the process is happening here. The major difference between these two is once the finished product is finished, that is protein or enzyme, to extract we have to remove the or we have to filter the whole broth. Okay, and second disadvantage is that there is no inlet and outlets are present here. So there is no sterile or the sterile steam or the sterile air can pass here, or there is no outlet to remove the downstreaming process, that is protein products which are finished later. So this is the difference between the bioreactor and the conical flask which you are using at the laboratory. And another disadvantage of conical flask is that, so once it is done, the process is done, we have to again go on inoculating the process by the microorganism. So this, I hope this clarifies here.
क्लास 6 से 12 से लेके नीट IIT जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज़्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।